Hi guys. Welcome to Easy Microbiology. We make microbiology easy. Today we will learn about the Staphylococcus aureus. 1. Definition of the genus. 2. Habitat. 3. Epidemiology. 4. Pathogenesis. 5. Virulence factors. 6. Clinical significance. 7. Laboratory diagnosis. 8. Biochemical test. 9. Treatment. Definition of the genus. Members of the genus Staphylococcus are gram-positive cocci that occur singly, in pairs, tetrads, short chains, and irregular grape-like clusters. They are non-motile and non-spore-forming. Most species demonstrate catalase activity and are facultative anaerobes, growing better under aerobic than anaerobic conditions. The anaerobic species Staphylococcus saccharolyticus and subspecies Staphylococcus aureus subspecies anaerobius are exceptions. Most species grow in the presence of 10% sodium chloride and at 18 to 40 degrees Celsius. Staphylococci are susceptible to furazolidone and resistant to low levels of bacitracin. They are susceptible to lysis by lysostaphin and are relatively resistant to lysis by lysozyme. The cell wall contains peptidoglycan and teichoic acid. The G and C content of DNA is 30 to 40 mol%. The estimated genome size of the members of the genus Staphylococcus species is of 2,000 to 3,000 kilobase pair. For more content and notes, visit our website at description below. Our website looks like as shown in the figure. Two. Habitat. Cutaneous habitats, excluding the nose. Staphylococci are one of the major groups of bacteria inhabiting the skin, skin glands, and mucous membranes of mammals. The commonest resident staphylococcal species on human skin in terms of population size is Staphylococcus epidermidis. The anterior nares. The nose provides a major habitat for Staphylococcus epidermidis, and is the dominant ecological niche for Staphylococcus aureus. Three. Epidemiology. Staphylococcus aureus is frequently carried by healthy individuals on the skin and mucous membranes. Carriers serve as a source of infection to themselves and others. 4. Pathogenesis. As shown in figure. Five. What is virulence factor? Virulence factors are the genetic, biochemical, or structural features that enable an organism to produce disease. The clinical outcome of an infection depends on the virulence of the pathogen and the opposing effectiveness of the host defense mechanisms. Cell wall virulence factors. Capsule. Most clinical isolates express a polysaccharide microcapsule of types 5 or 8. The capsule layer is very thin but has been associated with increased resistance to phagocytosis. Protein A. Protein A is a major component of the S. aureus cell wall. It binds to the Fc region of IgG, exerting an antiopsinin effect. Fibronectin binding protein. Fibronectin binding protein promote binding to mucosal cells and tissue matrices. Clumping factor. This FNBP enhances clumping of the organisms in the presence of plasma. Cytolytic exotoxins. Alpha, beta, gamma, and delta toxins attack mammalian cell membranes. It is chromosomally encoded. It polymerizes into tubes that pierce membranes, resulting in the loss of important molecules and, eventually, in osmotic lysis. Panton-Valentine leucocidin. This pore-forming toxin lyses PMN's production of this toxin makes strains more virulent. This toxin is produced predominantly by community-acquired methicillin-resistant S. aureus strains. Superantigen exotoxins. These toxins have an affinity for the TSL receptor, major histocompatibility complex class II antigen complex. They stimulate enhanced T lymphocyte response. Enterotoxins. Enterotoxins, 6 major antigenic types, A, B, C, D, E, and G, are produced by approximately half of all S. aureus isolates. When these bacteria contaminate food, and are allowed to grow, they secrete enterotoxin, ingestion of which can cause food poisoning. Toxic shock syndrome toxin, TSST, 1. This is the classic cause of toxic shock syndrome, TSS. Because of similarities in molecular structure, it is sometimes referred to as staphylococcal enterotoxin F, although it does not cause food poisoning when ingested. Exfoliatin, exfoliative toxin, ET, is also a superantigen. It causes scalded skin syndrome in children. The toxin cleaves desmoglein 1, which is a component of desmosomes. Cleavage results in loss of the superficial skin layer. (6) Clinical significance. Localized skin infections. Deep localized infections. Acute endocarditis. Septicemia. Pneumonia. Nosocomial infections. Toxinoses. Localized skin infections. The most common S. aureus infections are small, superficial abscesses involving hair follicles, folliculitis, or sweat or sebaceous glands. For example, the common STI, external hordeolum, is created by infection of an eyelash follicle. Subcutaneous abscesses called furuncles, boils, often form around foreign bodies such as splinters. Deep, localized infections. These may be metastatic from superficial infections or skin carriage or may result from trauma. S. aureus is the most common cause of acute and chronic infection of bone marrow. S. aureus is also the most common cause of acute infection of joint space in children, septic joint. Acute endocarditis, generally associated with intravenous drug abuse. Acute endocarditis is caused by injection of contaminated preparations or by needles contaminated with S. aureus. Progression to septicemia is often a terminal event. Toxinoses, these are diseases caused by the action of a toxin, frequently when the organism that secreted the toxin is undetectable. Toxinoses caused by S. aureus include toxic shock syndrome, TSS results in high fever, rash, vomiting, diarrhea, hypotension, and multi-organ involvement. This toxin is responsible for toxic shock syndrome, TSS. It has two subtypes, TSST1 and TSST2. TSST1 is actually a staphylococcal enterotoxin. Enterotoxin F or pyrogenic exotoxin C is most common type of TSST1. Rarely enterotoxin B or C may also be associated. Staphylococcal gastroenteritis, This is caused by ingestion of food contaminated with enterotoxin-producing S. aureus. Often contaminated by a food handler, these foods tend to be protein-rich or salty, like ham, and improperly refrigerated. These heat-resistant toxins are able to withstand subsequent reheating. Scalded skin syndrome. This involves the appearance of superficial bullae resulting from the action of an exfoliative toxin that attacks the intercellular adhesive of the stratum granulosum, causing marked epithelial desquamation. The bullae may be infected or may result from toxin produced by organisms infecting a different site. Laboratory diagnosis. Sample collection. It depends on the nature of the lesion. Direct smear microscopy. Gram staining of pus or wound swab reveals pus cells with gram-positive cocci in clusters. However, direct microscopy is of no value when S. aureus is a part of normal flora in the sample. Culture. Specimens are inoculated on various media and incubated overnight at 37 degrees Celsius aerobically. The colony morphology is observed as follows. Nutrient agar. Colonies are 1-3 mm in size, circular, smooth, convex, opaque and easily emulsifiable. Most strains produce golden yellow non-diffusible pigments. Grown anaerobically, colonies are smaller and grayish in color. Nutrient agar slope. It produces confluent growth. Blood agar. Colonies are similar to that on nutrient agar, in addition surrounded by a narrow zone of beta hemolysis McConkey agar. Small pink colonies are produced due to lactose fermentation. Liquid medium. It produces uniform turbidity. Selective medium mannitol sale agar contains nutrient agar with 7.5% NaCl and phenol red as an indicator. Salt milk agar contains nutrient agar, 6.5% NaCl and I0% skimmed milk. Biochemical test. Catalase positive. Citrate positive. Coagulase positive. Gas negative. Gelatin hydrolysis positive. Gram staining positive. H2S negative. Hemolysis positive beta. Indole negative. MR, methyl red, positive. Nitrate reduction positive. Of fermentative. Oxidase negative. Urease positive. VP positive. Alkaline phosphatase positive. Arginine dehydrolase positive. For detailed knowledge and notes visit Easy Microbiology from the description.